There's not much metering Dr. Kasper Roet's confidence in his company's technology can conquer neurological disorders like ALS if his longstanding and hands-on commitment to neuro research is any indication. That confidence is well placed. I'm Ben Comer.
And I'm Matt Pillar.
And this is the Business of Biotech, JP Morgan 2025 edition. And we're here in San Francisco at the law offices of Alston and Bird with Dr. Roet, founder and CEO of QurAlis, to explore the why behind his optimism. Dr. Roet, thanks for being here.
My pleasure. Thanks for inviting me.
It's our pleasure, Dr. Roet. And I noticed you rolled the R on the first pronunciation and then rolled right back into the straight American.
Yeah, I tried it and I thought it sounded a little funny coming out of my mouth. I need to practice it some more before I pull it back out.
I give you kudos for even trying.
I appreciate that.
So you've got a very diverse and hands-on background in pretty much everything that you've done, and I'm curious about the beginnings of your career. Before you got into life sciences, you started your career in IT. Is that correct?
That is correct.
Yeah,
Matt.
So tell us a little bit about that and where, when, and why the pivot from IT into life sciences occurred.
My IT life comes from the fact that I have a passion for computers. I started that as a very young child, and when the internet boom started in the 1990s, I was one of the few people that knew how to assemble computers, take them apart, program them, build Wi-Fi networks. And so when I was finishing up my bachelor degree at the University of Amsterdam, me and two friends were asked to help with small companies and student houses to set up their IT infrastructure so that everyone could get access to the internet. So I kind of rolled that into a company, and that became actually an IT company that first built infrastructure, then built web applications, and then also placed IT specialists at other companies to solve their problems. I had never anticipated being an entrepreneur before that. It was kind of a natural evolution to become an entrepreneur. And when I realized how great it was to actually be able to create your own visions into reality, I was caught. And I knew I wanted to be an entrepreneur. But I also knew I didn't actually want to be an IT entrepreneur. So my passion had always been on biology. And I really wanted to develop medicine so that we could actually have a world, hopefully a future world where we don't suffer from diseases anymore.
Yeah. Well, let me just say that I think you made the right choice because your personality lends itself to medicine. I covered IT for like 20 years, and the IT space, the people that I interviewed in the IT space, not nearly as chill as you are, right? Like there's bigger egos in the IT space than there are in the life sciences. So I think you made the right decision based on what I know about you so far.
Good.
What was your first computer?
My first computer, like I had a Commodore 64, but I actually had like a tape game computer before that. I don't remember what it was called anymore. But I think I guess I'm an Atari before the Commodore. So it's like evolved.
And you took all those apart?
Yes. Yeah. I always tried to make them a little faster and crank up their memory a little bit so I could play better games. That was really the big motivator.
Yeah. What was your foray when you decided that you wanted to pursue something in the life sciences, medicine related? What was your first foray into that field?
My first foray, when I started actually with artificial intelligence as in my university track. But I come from the fields of a small farm town. Actually, the first lake that was dried out, the big lake in the Netherlands, was where I grew up. And I had a microscope and I caught little bugs in the field, and I was just taking them apart to see how they work. And that was really a passion for me. And it was not just taking the bugs apart, but understanding biology and understanding nature. And so I was doing this artificial intelligence, but I realized other people are probably better at programming. And I realized I should just do what I'm really good at, and decided to study medical biology again at the University of Amsterdam. And I loved it. The moment I started that, I was just in love with it. And then after that, got rolled in this IT company, but I went back and did a master's in neuroscience. And there, I was working in one of the foremost gene therapy labs in the Netherlands, the lab of Joost Verhagen, was really pushing gene therapy technologies forward because I wanted to translate. At that moment, I already knew I wanted to make medicine, and how gene therapy is actually an ultimate medicine. And that kind of got me the exposure in the neuroscience space, because that is where Joost was working to make gene therapies for spinal cord repair for paralyzed people. And so I did my master's internship there. And then I did a master's internship in UCSD with Mark Tuschinski, a big gene therapy scientist there. And then I came back to do a PhD, but again, I wanted to do something that was really translational. So I started a gene therapy PhD project, which was a collaboration between the University of Amsterdam, the Free University, AMT, which was Amsterdam Molecular Therapeutics, which later became Unicure. So quite a successful gene therapy company. At that time, they actually brought the first gene therapy into the Western markets for Cloudbera. And so I was there at that time while they were doing that, and I was also collaborating with Galapagos. They were part of the project. Basically, it was called the Center Novum project, again, to bring gene therapies to spinal cord repair. And so the institute where I worked, the Netherlands Institute for Neuroscience, was actually the director there started the Dutch Brain Bank. And the Dutch Brain Bank is one of the best brain banks in the world, because usually within 2 hours after a donor passes away, the body is brought to the hospital, and we're following an autopsy protocol to take out brain and spinal cord and process that in a certain way, depending on the disease. And I started to work there and became an autopsy coordinator, which meant that in the middle of the night, I would get a phone call, someone has passed away. Can you please organize the autopsy? So I would call the families, I would call the undertaker, I would call the neuropathologist, the section assistant. We would all go to the hospital. We would receive the body. We would then look at it, and I could see the impact that all these neurodegenerative diseases have on patients, talking to their family, looking at their bodies, looking at their brains, looking at their spinal cords, taking it apart, and sometimes bringing the tissues back to the institute, where sometimes in the middle of the night, researchers would come to try to find out things that were happening in those diseases. And that really caught me into the broader neurodegenerative space. And so, that is what really started to motivate me, just the incredible impact that those diseases have on those patients, both on them, obviously they've passed away at that point, but also on their families. And I decided I do and really specialized in development of therapies for neurodegenerative diseases.
How did that really hands-on experience at the Netherlands Brain Bank influence your career and where you are now? Because that strikes me as unique. Not everyone in the industry has that degree of handling a brain that works in neuroscience.
Yeah, I can tell you it's really helpful right now, because we're working on an autopsy program for our current trial, and I understand how it works, what it means to have tissue, how it's processed. I'm the CEO, but it's good to have that type of understanding. We're actually, my company is now collaborating with the Dutch Brain Bank. We just started a program also for Fragile X. We wanted to get tissue from Fragile X patients, and the tissue that we got from these patients, we've been able to study and really confirm a genetic target for Fragile X. I mean, that has really helped me to appreciate the tissue. One of the other things that I really took away from that is how often patients are misdiagnosed, because clinicians misdiagnose patients based on clinical symptoms. But once you open up someone's body and you look actually on, how the disease looks inside the body, very often we saw that the clinical diagnosis was not correct. But a clinical diagnosis often stands. And so when you get tissue from a donor, the clinical diagnosis is the diagnosis. You have to really read through that to see if the pathological diagnosis is the same, because otherwise you're actually drawing the wrong conclusions on the materials that you use. And that's a very practical point where my experience there has really given me caution on interpretation of data from material from donors. And I think it's helping us do better things in QurAlis.
Yeah. We kind of fast forwarded to QurAlis. There were a couple of stops between the Dutch Brain Bank and your founding of QurAlis.
Yeah. Again, I knew I wanted to develop medicine, and I realized I know how a lot of companies worked. I worked as a computer specialist in a computer company as a side job. I was a telephone marketer for a while. I worked at an investment bank. And so I kind of knew a lot of different sectors, worked in a bar, had no idea how you make a drug. And it's like, if I want to start a company developing therapeutics, I really need to understand how to make a drug. And so in the Netherlands, one of the big R&D organizations at that moment there was J&J, who had their Janssen vaccine site there, which was formerly QurAlis, but was acquired by J&J just before that time that I graduated with my PhD. And I applied there and got a job first as an assay potency development team leader, and that kind of progressed into becoming a technical integrator. And there I learned how you get something from research all the way to the clinic, because I work together with formulation, fill and finish, upstream processing, downstream processing, clinical assays, QC assays, R&D, CMC. And there, a couple of years in, I really understood how you can actually make a drug and how the pharmaceutical business works and what type of experts you need and all of that. And while I was doing that, C9orf72 was discovered, big ALS gene. My PhD program was spinal cord repair gene therapies, so neuromuscular. ALS is a neuromuscular disease, and so it's the same tissues, but I thought we cannot treat ALS because we don't understand the disease. But while I was at J&J, a big ALS gene was discovered, and then rapidly 38 other ALS genes were discovered. And I'm a big data person, probably because of my passion for computers. And so I started looking at that, and I thought, this is incredible. We have the genetics of the disease. I could see pathways emerge. We could see disease mechanisms emerge. And so in my hobby time, I took published data sets from postmortem tissues, again, connected to my work at the Dutch Brain Bank, looked at all the transcriptional profiles in a whole genome way to identify hub genes that could explain why the disease actually develops and how you could potentially treat it. I also realized we need a translational model so that we can test therapeutics and test hypotheses, and did the same with all of the animal models that were published, all of the transcription data sets from all of the different ALS models that were published. So I built this whole meta-analysis of human and animal we'll call it postmortem tissue, to see if I could find things that were conserved between the two model systems. Found zero overlap. I was incredibly frustrated because we have the genetics, we know what's gone wrong in patients, we can probably develop therapeutics, but we have no way to test it. How do we test this? And then I discovered Kevin Eggen's work, who was at Harvard and had taken skin cells from a patient together with his collaborator at Mass General Hospital, James Berry. And he had used the Yamanaka protocols to turn those into stem cells. And he's a developmental biologist by training and knew how to make motor neurons from stem cells. And so he was able to create motor neurons from patients. And so the same neurons that are dying in a patient's spinal cord, you could recreate from a skin cell and study. And so I thought, this is phenomenal. That must translate. If we can see what is going wrong in a patient's own cells, in the cells that are dying in their disease, and we can test potential therapeutics, then it must be translating again back to patients. And so I decided to quit my job at J&J. I looked up Kevin, who was collaborating with Clifford Wolf, another professor, the head of the neurobiology center at Children's Hospital. And I said, can I please work with you? Because I want to learn this technology, stem cell technology, which was super controversial at that time. They liked my industry background. They had a collaboration with GSK. I had no one in their labs who actually knew how to work with pharma companies. So it kind of was a good match. And then for 3 years, I worked together with them, learned everything I wanted to know about these human neuronal model systems. And then I decided I'm done. I'm going to start my company. And so I told Kevin, and my plan was to go back to Europe to do that. So I told Kevin, I'm like, thank you so much. It's been incredibly great experience. I'm going to start an ALS company. And I talked to him about that before, and he said, oh, maybe we can do something together. And I never really took him serious. He was a Harvard professor. But then he said, well, let's do that together. And let's do it here. Let me introduce you to some people who might be interested in being part of that. And that actually led to the creation of QurAlis. And both Kevin and Clifford became co-founders together with myself and the CEO of another company that Kevin started, Jonathan Fleming, who used to be a managing partner at Oxford Biosciences. He decided to be part of that as well. And we created QurAlis to develop precision therapeutics for ALS based on the insight derived from these patient-derived disease models. And so the first two big discoveries that were made are now in the clinic. Those are our first clinical programs.
I had just a follow-up on J&J. Is QurAlis a member of J Labs, part of the incubator there?
Yes.
And what sort of benefits come along with that? What's your experience with that program?
I think J&J has J Labs to get close access to new and exciting technologies. So, QurAlis started, so the company where Jonathan worked was called QState, and QurAlis was founded out of QState. So QState was kind of helping get QurAlis established. The first office that I had was basically a closet in QState. It's like a little room in the middle of the building without any windows. My wife bought me a plant. The plant was the real employee in the office. But we were raising a little bit of seed money, and then someone advised me to apply for a golden ticket with Amgen to get access to Lab Central. Lab Central is one of the maybe the foremost bio-incubator space in the world. It's like in the middle of Kendall Square, 700 Main Street, started by Johannes Fruhauf, and I won. QurAlis won the Amgen Golden Ticket. I have a ticket like this big,
Yeah,
like a piece of cardboard, but it gave me access to Lab Central, and I actually didn't know what it was until I got there. J Labs is a sponsor of Lab Central. And so our office that we got at Lab Central was right next to J Labs office. And so suddenly we had this, and then we became a J Labs member. We applied and we won that as well. But what you then get is a very close vicinity to a large pharmaceutical company and the people that are interested in new technologies. We were bringing precision therapies based on stem cell technology, which is a very new technology at that time. J&J was very interested in that. The other company that was right next to that was Roche. They also had an office like right next to us. And so that is one of these amazing things that you have in Kendall Square. You're just close to all of these companies. And we won another golden ticket from Pfizer, got another year of extension at Lab Central. But it's really the proximity of small companies to large companies, which makes Cambridge and Massachusetts such an exciting environment, basically. And Lab Central is kind of pinnacle of all of that, because that's where it all kind of comes together in a very close density.
Where's the company located now?
So at some point when we were starting to close our Series A, it was time for us to get more space. I wanted to stay in Kendall because I like the buzz, but my team said, Kasper, the commute to Kendall is really painful for us. We live outside of the city. It takes us half an hour just to be in Cambridge, and then half an hour back if it's a good day. So can we get a little bit further away? So I said, fine, but we're going to stay in Cambridge. So we are now behind the Ilweg station in West Cambridge. There's a little biotech hub there as well. GSK sits there, UCB sits there, ASI sits there, and a bunch of little companies. And it's actually quite a nice, a little small cabinet, basically.